You are welcome to my another session of cytology class. So the topic of discussion of this session is cell division with respect to meiosis II. At the same time, at the end, we also have to discuss about the similarities and differences between the two main types of cell divisions, that is mitosis and meiosis. Meiosis II, also called as equational or homotypic division. Meiosis II is exactly like a mitosis, and therefore it is commonly called as meiotic mitosis. In this division, two haploid chromosomes split longitudinally and distributed equally to form four haploid cells. It completes in four different stages, and the stages are Prophase II, Metaphase II, Anaphase II, and lastly Telophase II. Prophase II. Here, the dyad's chromosomes becomes thicker and shorter. The nuclear membrane and nucleolus disappear. The spindle fiber starts to form. In case of metaphase II, the dyad chromosomes comes to equatorial plane. The spindle fibers organize between the poles and attaches to the centromere of the chromosomes. In anaphase II, the centromere of each chromosome divides and sister chromatids separates to form two daughter chromosomes. The spindle fiber contracts and pull the daughter chromosomes apart towards the opposite poles. In telophase II, the chromosome becomes organized at respective pole into nuclei. The chromosomes elongates to form a thin network of chromatin. The nuclear membrane and nucleolus reappears. Now cytokinesis 2. The result of cytokinesis is the formation of four haploid daughter cells, that is gametes or spores. The cytokinesis takes place by cell plate formation in plant cell. There are two methods by which it takes place. One is successive method where cytokinesis is followed by each nuclear division resulting in four haploid cells, for example in monocot plants. Whereas in another type, that is simultaneous method, cytokinesis occurs only after meiosis II to form four haploid cells, and the examples are dicot plants. But in animal cells, cytokinesis occurs by furrow formation or depression. These are the diagrammatic representations of meiosis, both meiosis I and meiosis II. Metaphase I, anaphase I, this is telophase I, then prophase II in meiosis II, and metaphase II, anaphase II, telophase II. So the end product of meiosis, formation of four haploid daughter cells. Now coming to the significance of meiosis. Meiosis helps to maintain a constant number of chromosomes by reducing the chromosome number in the gametes. The essential for sexual reproduction in higher animals and plants. Meiosis helps in the formation of haploid gametes and spores for sexual reproduction. Number of chromosomes remain fixed in a species from generation to generation. The crossing over occurring brings genetic variations in offspring, which helps in evolution of organisms. Failure disjunction in meiosis leads to mutation and ultimately leads to the formation of polyploid forms. The random distribution of maternal and paternal chromosomes takes place into daughter cells during meiosis, and it is a sort of independent assortment which leads to variations. Mitosis and meiosis, similarities and differences. Mitosis and meiosis are both the processes of cell division. Organisms are constantly replenishing their cell supply and creating new cells to replace those that are old or damaged, as well as making cells to be used to create new organisms during sexual reproduction. The mitosis is when a cell divides to create two identical daughter cells. Mitosis occurs in somatic cells, and it is a process critical for producing new cells and keeping the organism alive and healthy. All the cells go through the process of formation, growth, division, and eventual death. During mitosis, the parent cell nucleus is split to form two sets of chromosomes for which each of the new daughter cells. Meiosis also involves cell division. However, it is needed for sexual reproduction, and each cycle of meiosis creates four daughter cells with exactly half the number of chromosomes as parent cell. During fertilization, two daughter cells will combine to create an embryo with a full set of chromosomes. Below is an information which gives some similarities and differences between these two cell divisions. So this is a table which gives a comparative account between the mitosis and meiosis. So character, mitotic division and meiotic division. So alternate name, equational division, here it is reducing division. Discovered by Walter Fleming, and here it is Oscar Hartwig. The type of cells involved are somatic cells, and here it is sex cells or germ cells. Then type of reproduction, it is asexual, whereas it is sexual. Mother cells involved can be either haploid or diploid, and here it is always diploid. The number of divisions, mitosis involves only one cell division, and here it involves two successive divisions, that is meiosis I and meiosis II. As far as duration is concerned, comparatively it is shorter, and here it is longer. Then number of daughter cells at the end in mitosis 2, in meiosis 4. Reduction in chromosome number, here it is no reduction. In meiosis results in the reduction in number by half. As far as genetic identity is concerned, daughter cells are genetically same, but here daughter cells are genetically different due to recombination. So as far as recombination and crossing over is concerned, mitosis, no, nothing like that, and whereas meiosis, crossing over occurs. And steps involved: prophase, metaphase, anaphase, telophase. Whereas here in meiosis 1 and meiosis 2, meiosis 1 with prophase 1, metaphase 1, anaphase 1, telophase 1, and meiosis 2 with prophase 2, metaphase 2, anaphase 2, and telophase 2. The interphase occurs prior to each division, and interphase proceeds in only meiosis 1. It does not occur prior to meiosis 2. DNA replication takes place during interphase in mitosis, and it takes place during interphase 1 but not during interphase 2 in meiosis. As far prophase is concerned, cell spends less time in prophase and no substages are there in mitosis. And in meiosis, prophase 1 consists of 5 substages like leptotene, zygotene, pachytene, diplotene and diakinesis. Synapsis formation is absent, whereas in meiosis it is there. It occurs between the two homologous chromosomes during prophase. In tetrad formation, absent in mitosis, a tetrad consisting of 4 chromatids, that is 2 sets of sister chromatids lined up closely together, it is formed in meiosis. Then the chiasma, absent in mitosis, observed during prophase 1 of meiosis. The centromere, each chromosome consists of 2 chromatids united by a centromere. In meiosis, 2 homologous chromosomes form bivalents or tetrads. Each bivalent has 4 chromatids and 2 centromeres. In metaphase, sister chromatids align at the metaphase plate. All the centromeres line up in the same plate. Whereas in meiosis, tetrads align at the metaphase plate 1 in metaphase 1 and the centromeres are lined up in 2 planes which are parallel to one another. In anaphase, sister chromatids separate and begin migrating centromere first toward the opposite poles of the cell. A separated sister chromatid becomes a daughter chromosome. In anaphase 1, double-stranded chromosomes are separated towards each cellular pole. Sister chromatids do not separate. On the other hand, chromosomes are the ones being segregated during Anaphase II. Centromeres. The centromeres split during anaphase. The centromeres do not separate during anaphase 1 but takes place during anaphase 2. Cytokinesis occurs at the end of the telophase in mitosis. In meiosis, it happens at the end of telophase 1 and telophase 2. Functions. Mitosis helps in growth, repair and replacement and helps to produce more cells, especially during early stages of development, and also helps to regenerate damaged and lost cells. Whereas in meiosis, it takes part in the formation of gametes. It maintains the chromosome number of the offspring. Then for the maintenance of the genetic diversity on which the process of natural selection acts upon.